హలో ఆల్, ఈరోజు మనం చూడబోయే టాపిక్ ఏంటంటే ద వెరీ ఇంట్రెస్టింగ్ టాపిక్ ఫర్ సెకండ్ ఇయర్ ఎన్సిఆర్టీ, ప్రిన్సిపల్స్ ఆఫ్ ఇన్హెరిటెన్స్. ప్రిన్సిపల్స్ ఆఫ్ ఇన్హెరిటెన్స్ అండ్ వేరియేషన్. ఓకే, సో ఈరోజు ఈ టాపిక్ కవర్ చేయబోతున్నాం. ఈ టాపిక్లో మనకి యూజువల్గా ఏంటంటే, ఈ టాపిక్లో చాలా కన్ఫ్యూజింగ్ పార్ట్ ఏంటంటే, హెరిడిటరీ అంటే ఏంటి, ఇన్హెరిటెన్స్ అంటే ఏంటి, వేరియేషన్ అంటే ఏంటి, జీన్ అంటే ఏంటి, అల్లీల్ అంటే ఏంటి, జీనోటైప్ అంటే ఏంటి, ఫినోటైప్ అంటే ఏంటో డీటెయిల్డ్గా చూద్దాం. సో వాట్ ఈస్ జెనెటిక్స్? Genetics is none other than a branch of biology which deals with hereditary. So a branch of biology which deals with, sorry, excuse me, in spelling tappu rasanu. Heredity and variation. Okay, so next is the term heredity is coined by, ఈ వర్డ్ని కాయిన్ చేసింది ఎవరంటే బీట్సన్. అండ్ హెరిడిటరీ, వాట్ ఈస్ హెరిడిటీ? హెరిడిటీ ఈజ్ నథింగ్ బట్ ఇట్ ఈస్ ఎ లాటిన్ వర్డ్. సో యూ కెన్ సీ ఇట్ ఈస్ ఎ లాటిన్ వర్డ్. అండ్ సో ద హెరిడిటమ్ అనే ఒక లాటిన్ వర్డ్ నుంచి ఇది తీశారు, అంటే బీయింగ్ ఏ హెయిర్ అని అర్థం. అండ్ ఇన్హెరిటెన్స్ ఇస్ ద ప్రాసెస్. ఇన్హెరిటెన్స్ అనేది ప్రాసెస్ అన్నమాట. హెరిడిటీ అంటే ట్రాన్స్ఫర్ ఆఫ్ ట్రైట్స్ ఫ్రమ్ వన్ జనరేషన్స్ టు నెక్స్ట్ జనరేషన్, అది హెరిడిటరీ. ట్రాన్స్ఫర్ ఆఫ్ ట్రైట్స్ ఫ్రమ్ వన్ జనరేషన్ టు నెక్స్ట్ జనరేషన్, దీన్ని మనం హెరిడిటరీ అని పిలుస్తాం. అదే ఇన్హెరిటెన్స్ అంటే ఏంటంటే ద ప్రాసెస్ ఆఫ్ క్యారెక్టరిస్టిక్స్ ఈజ్ పాస్డ్ ఫ్రమ్ పేరెంట్ టు ప్రోజెనీ. సో ప్రాసెస్ ఆఫ్ ట్రాన్స్ఫర్ బై విచ్, ప్రాసెస్ని ఇన్హెరిటెన్స్ అంటాము. సో ట్రాన్స్ఫరింగ్ ఆఫ్ ట్రైట్స్ ఫ్రమ్ వన్ జనరేషన్స్ టు నెక్స్ట్ జనరేషన్ని హెరిడిటరీ అంటాం. హెరిడిటరీ అంటే ఏం మనకి వచ్చాయి అని, ఇన్హెరిటెన్స్ అంటే ప్రాసెస్ బై విచ్ characteristics are passed from one generation to progeny. What is variation? Degree by which the progeny will differ from their parent. What is variation? Degree by which the progeny differs from parent. సో దీన్ని మనం ఏమంటామంటే వేరియేషన్ అంటాం. సో హెరిడిటరీ, ఇన్హెరిటెన్స్, వేరియేషన్. హెరిడిటరీ అంటే ఏంటంటే త్రూ విచ్, సో ద ట్రైట్స్ విచ్ ఈస్ పాస్డ్ ఫ్రమ్ వన్ జనరేషన్ టు నెక్స్ట్ జనరేషన్. ద ప్రాసెస్ ఆఫ్ ట్రాన్స్ఫర్ బై విచ్ ద క్యారెక్టరిస్టిక్ ఫ్యూచర్స్ ఈస్ పాస్డ్ ఫ్రమ్ వన్ జనరేషన్ టు నెక్స్ట్ జనరేషన్, దాన్ని మనం ఇన్హెరిటెన్స్ అంటాం. వేరియేషన్ అంటే ఏంటంటే ద డిగ్రీ బై విచ్ ద ప్రోజనీ డిఫర్స్ ఫ్రమ్ ద పేరెంట్. And next, for thousands of years, humans have been selectively breeding both plants and animals, so to produce useful hybrids. So what is the meaning of hybrid? So offsprings we will be getting offsprings by crossing the parent of different characteristics. Like for example, tall is crossed with dwarf. Violet flowers is crossed with white flowers. సో దీన్నే మనం హైబ్రిడ్ అంటాం. సో మనకి ద బెస్ట్ ఎగ్జాంపుల్ వచ్చి వెల్ నోన్ ఇండియన్ బ్రీడ్ సహీవాల్. cattle was developed in Punjab, and genetic improvement was based on controlling breeding. అంటే మనకి ఏం క్యారెక్టర్స్ కావాలో వాటి వరకే. సో యాక్చువల్ మెకానిజం ఏంటంటే ఆఫ్ ఇన్హెరిటెన్స్ వాజ్ అన్నోన్. సో ఈ నాలెడ్జ్ ఆఫ్ జెనెటిక్ మెకానిజం ఏంటంటే ఫైనలీ కేమ్ టు ద రిజల్ట్ బై ఎక్స్పెరిమెంటింగ్. మనకి ఇంతకుముందు ఇలా సెలెక్టివ్ బ్రీడింగ్ వల్ల క్యారెక్టర్స్ వస్తాయని తెలుసు, కానీ ఎలానో ప్రాసెస్ తెలీదు. సో లేటర్గా ఏంటంటే జెనెటిక్ మెకానిజమ్స్ ఫైనల్గా మనకి తెలిసాయన్నమాట చాలా ఎక్స్పెరిమెంట్స్ చేయడం వల్ల. సో ఈ చాప్టర్లో ఏంటంటే లాస్ ఆఫ్ హెరిడిటీ అండ్ వేరియేషన్ అండ్ అలీలిక్ ఇంటరాక్షన్స్, ఇన్హెరిటెన్స్ ఆఫ్ జీన్, సెక్స్ డిటర్మినేషన్, ఇంకా జెనెటిక్ డిసార్డర్స్ గురించి చూస్తాం. సో వాట్ ఈస్ ద స్పెషాలిటీ ఆఫ్ సహీవాల్ కవ్? సహీవాల్ కవ్ ఏంటంటే ఇది చాలా పెద్ద అడ్డర్ కలిగి ఉంటుంది అండ్ ఇవి ఎక్కువ పాలు ఇవ్వగలవు. ప్లస్ అట్ ద సేమ్ టైం ఏంటంటే డిసీజ్ రెసిస్టెంట్ కూడా, అండ్ చాలా లాంగిటివిటీ ఉంటుందన్నమాట దీనికి లైఫ్ టైం కూడా. సో అందుకని సహీవాల్ అనేది చాలా ఫేమస్ హైబ్రిడ్ విచ్ ఈస్ డెవలప్డ్ ఇన్ పంజాబ్. నెక్స్ట్, వాట్ ఈస్ జీన్? సో జీన్ అంటే ఏంటి? జీన్ ఈజ్ నన్ అదర్ దాన్ యూనిట్ ఆఫ్ heredity. So unit of heredity we call it as gene, a unit of genetic information that occupies a specific position on the chromosomes. Chromosomes మీద ఇవి ఒక స్పెసిఫిక్ పొజిషన్లో ఉంటాయి. ఫర్ ఎగ్జాంపుల్, సో ఇది క్రోమోజోమ్గా తీసుకుంటే ఇక్కడ ఒక జీన్స్ ఉంటాయి, ఇక్కడ ఒక జీన్స్ ఉంటాయి, ఇక్కడ ఒక జీన్స్ ఉంటాయి, ఇలాగా. సో జీన్స్ అనేవి నన్ అదర్ దాన్ ఇవి ఒక జెనెటిక్ ఇన్ఫర్మేషన్. అవి క్రోమోజోమ్స్ మీద స్పెసిఫిక్ పొజిషన్ని ఆక్యుపై చేస్తాయి. ఈ పొజిషన్ని మనం ఏమంటాం అంటే లోకై అంటాం. సో ఈ టర్మ్ని కాయిన్ చేసిన జెనెసిస్ట్ వచ్చి జోహాన్సన్ కాయిన్ ద టర్మ్ జీన్. సో బేస్డ్ ఆన్ ద గ్రీక్ వర్డ్ జెన్ అంటే రేస్ లేదా ఆర్ కైండ్. And next, what is allele? Allele means the various forms of genes or the varieties of genes, we call it as alleles. The varieties of genes we call it as alleles. For example, for height, capital T, small t. For flower, capital W, small w, flower color. So likewise for blood groups, IA, IB, IO. So like this, these are the allelic forms of blood groups. These are the allelic form for stem height. These are the allelic forms of flower color. And next, homozygous. In which both the allelic forms are similar. దెన్ ఈ కండిషన్ని హోమోజైగస్ అంటాం. ఇది ప్యూర్ బ్రీడింగ్ వల్ల వస్తుంది. ఫర్ ఎగ్జాంపుల్ క్యాపిటల్ టీ, స్మాల్ టీ, క్యాపిటల్ డబ్ల్యూ, స్మాల్ డబ్ల్యూ, అండ్ హెటిరోజైగస్ వెన్ టూ allelic forms are different. So that we call it as heterozygous. So like capital T, small t, capital W, small w, and we have IA, IB. So these are heterozygous thing. And next, coming to the other thing called phenotype. The description of the genotype we call it as phenotype, or the observable characters of an organism. For example, capital T, the phenotype is tall. Small t, the phenotype is dwarf. Capital T, the phenotype is tall. So this is called phenotype. which means none other than the describing thing or the observable characters of the organism. Next, genotype. Genotype is the genetic makeup of the organism or the genetic composition of the organism. So, genetic type is, genotype is capital T, small t. This is genotype. Capital T, capital T. This is genotype. And capital W, small w. This is genotype. And both phenotype, genotype and gene was the term coined by Johansen. Okay. And next, coming to the main topic, Mendel's law of inheritance. So what is inheritance? The process by which the characteristics are passed from one generation to another generation. The number of hypotheses was suggested for hereditary from Hippocrates. Hippocrates, he is the father of medicine, who is a Greek philosopher. Hippocrates to Aristotle. Okay, so the number of things was being found out by him from Hippocrates to Aristotle, among others. But Gregor John Mendel, an Austrian monk, Gregor John Mendel, who is an Austrian monk, was the first person to formulate the law for governing inheritance based on his experiments. His ideas was being published by the year 1865, and he has conducted the experiments during 1856 to 1863. 7 years he worked for hereditary inheritance. So, and also he published his work in 1865, which is unrecognized until 1900. So, which was very long after his death. Till date, Mendel is recognized as father of genetics. Right. And next, while Mendel researching on the plant genetics through experiments, so the principles of hereditary, and he discovered apply to animals too, because the mechanisms of hereditary are same for all the complex forms, because the same principles we can apply to animals also. Why did Mendel was worked on? So though Mendel has been selective crossbreeding, what Mendel done is selective crossbreeding. Of garden pea. So here I have selected a plant of garden pea, which is Pisum sativum. Over many generations, so Mendel proposed 3 laws of inheritance. How many laws was being proposed by Mendel is 3 laws of inheritance was proposed by Mendel. And why did he choose this pea plant for his experiment? They can cultivate very easily. So easy cultivation. And requirement of minimum space and time. The plants possess number of externally visible characters. And having a very short lifespan. The flowers are cleistogamous. And it is possible to conduct the cross pollination by transferring the pollen grains from one flower to another. So easy cross pollination. So large number of seeds are provided. And next, so Mendel observed these 7 traits, observed the 7 traits and are easily recognizable and occurred as distinctly visible characters. He occurred 7 different characters, yeah. So he used that to study the inheritance of a single trait. So when he studied about a single trait, then he named it as monohybrid cross. Dihybrid cross, when he studied 2 different traits at the same time. And we have trihybrid cross also, 3 different traits at the same time. So the traits and their contrasting varieties are, let's see. So we have the characters. So contrasting characters. The very first thing is stem height. So contrastingly, one thing will be the dominant. You will be having the dominant. And the other thing is recessive. So coming to the dominant one, we have the tall, dwarf. And coming to the second one, stem height, flower position. Third one, flower color. And fourth one, seed color. Seed shape. Fruit color or pod color. Pod shape. So flower position will be of axillary is dominant and terminal is recessive. Flower color, violet is dominant, white is recessive. Seed color, yellow is dominant, green is recessive. Seed shape, round is dominant, wrinkled is recessive. Pod color, so this is pod actually, you can call it as fruit or pericarp also. Green is dominant here. You have to remember vice versa, seed color and pod color, and yellow is recessive. Pod shape, inflated is dominant, constricted is recessive. And next, prior to the experiments, Mendel created a pure breeding line. So let's see. Pure breed lines only he have selected. So for many generations which is been self-pollinated. So pure breeding plants are stable trait. So which you can see the homozygous thing. So from generations to generation crossing through self-pollination. So the Mendel crossed these contrasting characters. He planted the seeds to obtain the offsprings later. He then crossed the offsprings and he got the F2 generation. So let's see how to study one gene. So inheritance of one gene. So Mendel wished to know how character traits for the character may inherit and bond together. Among earlier experiment, Mendel has selected the stem height. Okay, so he have selected the parental variety. So tall cross to dwarf. So capital T, small t, small t. So capital T got only one type of gametes, small t gap one type. So all these things are capital T and small t. So later the Mendel's theory was being easily presented by Reginald C. Punnett by Punnett square. So capital T, capital T, capital T small t, capital T small t, capital T small t, and capital T small t. So all the genotypic ratio is 1:1:2, sorry, excuse me. One is to 1. And phenotypic ratio. Same, you will be having the same genotypic as well as phenotypic. Okay, so this is genotypic and phenotypic ratio. And coming to F2 generation, this is parental. This is genotype. This is the allele. And whatever we got is F1 generation. So let's see the F2 generation. F2 generation is none other than selfing the F1 generation. So capital T small t into capital T small t. Then again we'll go with Punnett square. Capital T small t, capital T small t, capital T, capital T, capital T small t, capital T small t, small t, small t. So this whatever you got is F2 generation, means Flyell2 generation. So in this the genotypic and phenotypic ratio will differ. So genotypic ratio is 1:2:1. You can see one type is so capital T and two types are capital T small t, and this one is so 1:2:1. And coming to the phenotypic ratio, 3:1 is the phenotypic ratio because you have the different thing. And next, this was okay. So now you can see this is selfing and self fertilization. So let's see what it is once again. So you have seen the parental generation of the pure lines. So they have taken pure lines, and this is genotype of a pure lines. They have crossed, and the allelic forms are this. And coming to the F1 generation, they got these resultant. All them are having same phenotypic and same genotypic, 1:1:1:1. Here both are same. When it comes to F1 generation is again self. So it leads to formation of F2 generation. So F2 generation, the genotypic and phenotypic ratios are different. Genotypic ratio is 1:2:1 and phenotypic ratio is 3:1. So let's see. So there was no intermediate forms. So he designated the plants with which the experiments were started as parental. We call that as P generation. Okay, parental or P generation. And the first hybrid he got is first Flyell. So in this, Philia is daughter. Phileus is son. Okay, so Flyell means offspring in Latin. So they got the F1 generation where all the plants are tall, and there will be inbreeding or selfing. So they'll went to F2 generation consisting of both tall and dwarf. In F2 generation, 75% are tall and 25% are dwarf. So the tall resembled the parents and grandparents, and dwarf resembled only the grandparents. There are no dwarf parents to them. There is no plants of intermediate height. You can see in a intermediate height. Appearance of the dwarfness in the F2 generation clearly showed that characters dwarfism have not disappeared. So they disappeared in F1 generation, but they reappeared in F2 generation. So the Mendel studied inheritance pattern of the remaining 6 characters also, like the same experiment, and he have the same results. Mendel used the letter like capital T for dominant. So capital T is dominant. Means uppercase is for dominant and small letters is for recessive. According to Mendel, the pure tall bleeding process, two identical, which is represented as capital T, and pure lines of dwarf is represented as identical factors small t. So, but during gametic formation, all the gametes will be having same type. So, if capital T, only capital T gametes will be there. If small t, only small t will be expressed. So, in the F1 generation, you got the offsprings, all are tall, but their genetical makeup is capital T and small t. So, when these are again selfed, the gametes has two different types of alleles. 50% of the gametes have capital T, 50% of the gametes have small t. So, that was the combination is there. And in the F2 generation, in the monohybrid cross, 70% of the offsprings are tall, 25% of the offsprings are dwarf, and the ratio is, you will be having the ratio of 1/4, so 1/2 and 1/4. So this is the ratio of genotype. So mathematically we'll be having a binomial expression as ax plus by square whole square. So gives rise to so they have converted the same half capital T plus half small t whole square gives rise to half capital T 1/2 capital T plus half small t into 1/3 capital T plus 1/2 small t gives rise to 1/4 capital T small t. Sorry, 1/4. 1/2. Excuse me. Capital T small t. Half capital T. Sorry. 1/4 capital T, 1/2 capital T small t, and 1/4 small t. Okay. So this is the equation, mathematical expression, binomial expression. And next, what is a test cross? So let's see what is a test cross. So what is a test cross is, here according to NCERT textbook we have a diagram. So they have given one flower. So here the flower can be of two ways: capital W, small w, capital W, capital W. So in both ways Here it is small w because both are homozygous recessive. So we will be drawing the Punnett square for the test cross. Here it is small, all are small w's. Here it is capital. Let me write you something bigger than these. Small w. This is also small w. So here you will be getting the flowers like capital W, small w, capital W, small w, capital W, small w, capital W, small w. So all are violet flowers. Then here the genotype is homozygous dominant. Here it is capital W and small w, capital W, small w, capital W, small w, small w, small w, small w, small w. In this case, 50% of the offsprings are violet. 50% of the offsprings are white, which means it is heterozygous dominant. Heterozygous dominant. Okay, so why we have to do a test cross is, this is test cross, in order to know the genotype of a plant. So because we cannot tell the genotype, genetic makeup we cannot tell, we can tell only the phenotype, right? So how do the Mendel know the genotype of a dominant phenotype? So a cross between a plant which is done showing the dominant phenotype. We have taken a violet flowers. It may be like homozygous or heterozygous. In both the case it will express as violet only. But we have to know the phenotype, right? So that's why. So what we have taken is, so in the test cross, the organism with dominant phenotype is crossed with recessive individual. In a monohybrid cross, the ratio will be 1:1, represents the offsprings. It means the parent with dominant phenotype is said to be heterozygous. On the other hand of the test cross, the parent is said to be homozygous. A test cross occurs only one pair of contrasting characters of the test cross. Okay, so in this case, we will be getting all 1:1. Here it is 2:2, which means 1:1. Coming to back cross. What is a back cross? Test cross is different and back cross is different. In a back cross, so which one, where the offspring is crossed either with one of the parent, hence test cross can be back cross, but all back crosses are not a test cross. So when the result of monohybrid cross is obtained, Mendel wondered if the results would vary if emasculation is performed on opposite parents than earlier. So thus he performed a reciprocal cross. So in the reciprocal cross of breeding experiment, to test the role of parental sex on inheritance, in one of the cross, the male expressing the trait of interest will be expressed with female expressing the contrast trait. In the test cross, for example, so with respect to height of the pea plant, if the tall parent has chosen as male in one set of cross, in the next, tall parent was emasculated, if reciprocal, that do not affect the phenotype of individual. This is understood that gender does not have any influence of the phenotype with back cross. Mendel performed the reciprocal crosses and depicted the same. Whereas if reciprocal crosses shows the gender influence, okay, so it is interfering with the gene as well as allosomes. So this was clearly referred in sex-linked inheritance. We will discuss later in that. So back cross is none other than crossing with parental type to see whether any sex influencing in inheritance. Next, Punnett square. Punnett square is none other than to calculate the probability of genotype. So it was developed by Punnett square. Developed by British geneticist called Reginald C. Punnett. The possible gametes are placed in vertical column representing the female, and horizontal row is male parent. So all possible combinations, vertical column is female, horizontal column is male. Okay, so the possible combinations will be put in the square. The Punnett square is nothing but the visual thing of Mendelian inheritance. So visual thing. Coming to the laws of inheritance. So we have two laws of inheritance when it comes to monohybrid cross. Two laws of inheritance only from monohybrid cross. We have two laws mainly. First law is law of dominance. It is very easy. The factors that is responsible for the contrasting traits stay together in the F1 generation, but they segregate during gametic formation in heterozygous. So it is based on the fact that the allele do not show any blending. So it was recovered. So the characters that is recovered in F1 generation, we have to call it as dominant. The characters which is not recovered in F1 generation, we will be calling it as recessive. And next we have the second law. So when it is expressed in F1 generation, it is dominant. When it is not expressed, you have to call it as recessive. Law of segregation. Law of segregation is whether in the in the parental generation both are homozygous. So all the gametes will be having same type of alleles. But when it comes to heterozygous, in the F2 generation, like in the F1 generation offsprings, the F1 generation offsprings will be having two different types, capital T and small t. When it comes to the gametes, 50% of the gametes will be having capital T and 50% of the gametes will be having small t. So this is going to tell us two different alleles segregate from each other. This is based on the fact that alleles do not show any blending. They will be recovered in F2 generation. So this is referred as concept of purity of genes. Concept of purity of genes. Purity of gametes. According to the concept of purity of gametes, so only the gamete will carry one allele. So that is regarded as one character. Other words, gametes said to be pure. Therefore, the law of purity of gametes. This is also, you can call it as law of segregation or law of purity of gametes. And next, we have the next concept, that is partial dominance or incomplete dominance. For this concept, it is not universal concept. It is not applicable to all, because the dominant gene do not completely express its dominant of a recessive. That's why it is incomplete dominance. So the F1 will show indeterminate. For this, they have selected the plant called dog flower. So we all call as snapdragon or Antirrhinum species. So the pure breed of red flower is selected. So red, the parental will be you can see the red flowers. At the same time, white flowers for the snapdragon. For the red flowers, the genotypic is given as capital R, capital R. So for the white flowers, small r, small r. So let's see the cross. This is parental or P generation. Now only same type of, only one type is. So let's see with the Punnett square. Capital R, small r, small r. So capital R, small r, capital R, small r, capital R, small r, and capital R, small r. This is F1 generation. So all are exhibiting pink flowers with phenotypic genotype. Phenotypic is 1:1:1:1. Genotypic is also 1:1:1:1. Okay, so this is F1 generation, and let's see the F2 generation. In the F2 generation, they crossed capital R, smaller, capital R, smaller. They crossed capital R, smaller, capital R, smaller, capital R, capital R, capital R, smaller, capital R, smaller, smaller, smaller. This is F2 generation. Coming to the phenotypic and genotypic ratio. Phenotypic is 1:2:1, and genotypic is also 1:2:1. In this incomplete dominance only, you will be getting like both the phenotypic and genotypic ratio is same for the F2 generation. So when pure breeding red flowers of the snapdragon is being crossed with pure breeding white flowers, all the F1 generation are pink flowers. So that means capital R is dominant. Capital R, red color is not able to dominate the recessive R. Where these pink color flowers are crossed among themselves, then the F2 generation produced 1:2:1. Same phenotypic and genotypic ratio they got. Okay, so blending cannot explain in such a way that red or white skipping a generation and pink flowers. So should produce only pink flowers. So flower colors in Mirabilis jalapa is also one more example of partial dominance. Mirabilis jalapa, 4 o'clock plant we call it as. So in snapdragon and Mirabilis jalapa is one more example. And coming to the concept of dominance here. So an allele doesn't has a capital letter or small letter. It exists the sequence of nitrogenous base. That is, inheritance patterns depends on how the individual, the gene, how the alleles of gene will interact to one another. Like in diploid organism, we have two alleles. Need not always should be identical. So in a normal case, normal enzyme which changes the substrate. So theoretically, the modified allele could be responsible for the production of less efficient enzyme, a normal enzyme, or a non-functional enzyme, or a no enzyme. For example, in first case, the modified allele will produce the same phenotypic that transforms the substrate into identical manner or lesser manner. So it produces a non-functional enzyme or no enzyme. The phenotype will be dependent upon the unmodified allele. The unmodified allele is generally considered as dominant and modified is generally as recessive. This is also possible for modified allele to produce completely new enzyme that is responsible for new trait. So coming to the discovery of Karl Korenz, a German of Germany. Karl Korenz of Germany. So through his work with 4 o'clock plant, Mirabilis jalapa. So Korenz replicated the same Mendel experiment in these plants. Korenz discovered the inheritance of variety called incomplete dominance. So incomplete dominance was discovered by Korenz. So let's see some of the things. Like how many pairs of contrasting characters studied by Mendel? 7. Among the 7 pairs of characters studied, number of traits related to flower, pod, seed. That is, for flower it is 2, for pod it is 2, for seed it is 2. Genes which code for a pair of contrasting traits is known as alleles. And what is antirrhinum is an example of incomplete dominance or partial dominance, both are same. And next, in monohybrid cross, 2 heterozygous individual, the percentage of pure homozygous individual in the offspring is 50% is homozygous, 50% is heterozygous. Next, the concept is regarding codominance. So an inheritance when the heterozygous individual will be having dissimilar or equally called codominance. For example, will be having both are expressing in this AB blood group system. So coming to the blood group, we have 4 different types of blood group: A, B, O, AB. Coming to the antigen on RBC, antibody in plasma. The nomenclature is based on antigen. Here it is antigen A, here it is antigen antibody B. Here it is antigen B, antibody A. Here there are no any antigens. Nil. Here both the antibodies are there, AB. Here the antigens are AB, here nil. So this is about the blood grouping system, what we follow. But when it comes to here, so this was discovered by Flix. Flix, who is a geneticist, explained the mechanism of ABO blood grouping system. So you have to remember the scientist called Felix Bernstein, the geneticist, explained the mechanism of inheritance of ABO blood group in the year 1924. So in the ABO blood group provides a good example for one more thing called multiple allelism. It is controlled by the gene called I. So abbreviated from isohemagglutinogen. So isohemagglutinogen. So which is located on the 9th chromosome. So they have 3 allelic forms, so which are 2 are found in an individual. So the gene responsible for antigen A is IA. The gene responsible for is IB. And the gene in which both antigens are absent, then it is IO. So the gene IO is recessive for IA and IB, and IA and B are codominants. If both are there, they both are expressed. So this phenomenon is an example of codominance. You can see the homozygous, heterozygous conditions also. So you can be expressed as, for example, assume that the blood groups and genotypes. A, B, AB, O. So here it can be IA, IA, or IA, IO, IB, IB, or IB, IO. Here it can be IA, IB. That's it only. Here it is IO, IO. So this is the way of expressing. So actually IA and IB will produce enzymes which attaches to specific sugar to chemical present on RBC membrane to form glycolipids actually. So but because it will produce antibodies. The RBC contain antigenic precursor also. So allelic IA is N-acetyl galactosamine transferase. Okay, so that's all about the codominance. So we have seen regarding the monohybrid cross, test cross, back cross, and we have seen the partial dominance, law of inheritance, partial dominance and codominance as well. So that's it. Thank you very much.